Peace be upon you all, and welcome to a new episode of Seeing God in Biology with Dr. Fuz Rana. Fuz, how are you doing today?
Ahmed, I'm doing great. How are you doing?
Always just fine. And today we have this mysterious topic, design of pseudogenes. So maybe first of all, pseudogenes looks like a topic that many people do not understand or do not know what it is. So I'm looking forward to your presentation.
Yeah, and just to get cut to the chase, and then we'll get into the nitty gritty. Is for many people, they view pseudogenes, which is a class of DNA sequences found in the genomes of organisms, as really being prima facie evidence for an evolutionary paradigm. The idea that all life, including humans, evolved through an unguided, undirected process. And so they see this as a direct challenge to the case that we've been making over the past several months for a creator's handiwork in the design of biochemical systems. And so oftentimes the response of people that have views like you and I have, where we see a creator, again, playing a critical role in the origin and the design of life, they oftentimes the response is really to operate on the defensive. And what I'm really going to argue here is that if we operate from a creation model or a design perspective, that we really should be on the offensive, where we see, pseudogenes as being an elegant part of the design of the human genome, and should prompt us to stand in awe before the creator and to worship and marvel at the creation's the creator's handiwork. And so that's kind of the mindset. But as you mentioned, probably many people aren't really familiar with pseudogenes. And so I need to give a little bit of background information, and then also talk a little bit about why many biologists see pseudogenes as being, again, evidence for an evolutionary history for life. So,
right, great. I guess many are now anxious to to get to it. Yeah.
Yeah, and to some degree, our story begins in June of 2000. Oh. And I don't know if you've ever seen this picture, Ahmed. It's a fairly famous picture, but this was taken in the Rose Garden of the White House. And of course, in the center is past President Bill Clinton. And on his left, on his right would be a person by the name of Craig Venter, who at that time was the president of a company called Celera Genomics. And on his left is Francis Collins, who was the head of the NIH's Human Genome Research Project. And both the Celera and NIH, along with other consortiums from other countries, were working to sequence the human genome, and they wound up in a race. And there was quite a bit at stake, because Celera Genomics was hoping to complete the human genome first and then commercialize the human genome sequence, selling people licenses to access that information. And Francis Collins and the public project was trying to sequence it and make that sequence data freely available to anyone who would want to make use of it. And so it became kind of a bitter scientific competition, a bitter rivalry formed between Venter and Collins. And somebody ended up mediating kind of an agreement between the two, where they agreed to share data with one another and complete a draft of the human genome, and then hold hands and walk across the finish line together, where no one really claimed any kind of victory. By the way, as an interesting aside, Craig Venter is an atheist and Francis Collins is an evangelical Christian, though he holds to kind of a form of theistic evolution. Yeah. But anyway, at that press conference, it was very interesting. Bill Clinton described the success of their work in this way, where he said that today we are learning the language in which God created life. we are gaining ever more awe for the complexity, the beauty, and the wonder of God's most divine and sacred gift. With this profound new knowledge, humankind is on the verge of gaining immense new power to heal. Genome science will have a real impact on all of our lives, and even more on the lives of our children. It will revolutionize the diagnosis, prevention, and treatment of most, if not all, human diseases. Now, I think this promise of the human genome is yet to be realized 20 years later. But what's very interesting to me is the religious language that Bill Clinton is using. And I think some of this, of course, is for political purposes, if I'm to be cynical. But nevertheless, I think you and I would agree with Bill Clinton that in the genome of organisms is really the language that God used to create life. And just for fun, here's a picture of me in the Wellcome Museum in London. And in the museum, they have actually a printout of the entire human genome. And it goes from floor to ceiling. So that's what 3.2 billion genetic letters listed in a sequence looks like. And the volumes go from 1 to 22, which corresponds to the 22 autosomes, and then there's an X and a Y volume. And they're organized from longest to shortest. So 1 is the longest sequence for a chromosome, and 22 is the shortest. But the picture doesn't do it justice, but the print is enormously small as it's listing out the letters. So that's a bit of just gives you a sense of the magnitude of the information in the human genome. And what's fascinating is that
there's like 30 to 40 trillion of those in our bodies, right?
Yeah, that's right.
It's unbelievable. It's really unbelievable when you see it.
And technically inside each cell, there's 6.4 billion genetic letters because each strand of DNA has a complementary sequence. So really there's 6.4 billion genetic letters. Now,
I was referring to the number of cells in the human body, and each of them would have like 30 to 40 trillion cells. And then each of them will have all this library in it, maybe multiplied by 2 because there's 2 copies in each.
Right. It's we could go on and on about how amazing it is. And of course, one of the first things that people wanted to do was determine how many genes are in the human genome. And to everybody's surprise, it was about 30,000. There are techniques that you can use to interrogate the sequences, looking for certain markers that indicate or connote the beginning of a gene sequence. But over time, people studied the composition of the human genome. And what people very quickly concluded is that only about 2% of the sequences in the human genome code for proteins. The rest appears to be non-coding DNA. And in fact, many of the sequences appear to be derived from retroviral infections like the line and sign and LTR sequences. And the bottom line is that most people viewed these sequences as non-functional, as junk. And so the argument at that time was that when we look at the human genome, we see anything but the language of God. In fact, it seems like it's the opposite. Why would a creator create this vast amount of DNA in the cells of every one of the cells of our body that is useless, that's non-functional? That just simply doesn't make sense. The evolutionary paradigm is the best way to make sense of this. And in fact, people argued that the structure of the human genome fits with what we would expect based on something known as the C-value paradox. And the C-value simply refers to the amount of DNA in the nucleus of gametes, like sperm cells and egg cells. And so it would be, in a sense, roughly corresponding to the size of what's called the haploid genome. Remember, because one part of our genome comes from our mother, one from the father. And so that's a total of 2 sets of of 23 chromosomes, and so half of that, or one set, is called the haploid genome, which is found in gametes. And the C value was something that was measured and people studied in earnest in the early 1970s. And at that time, again, the C value roughly corresponds to the size of the haploid genome. At that time, people thought, well, there should be a one-to-one relationship between the C value and the complexity of the organism, because more complex organisms would require more DNA. And lo and behold, when people looked at the C value, they noted that the C value was the same for every cell in an organism's body, but from organism to organism, the C value was all over the place. It had no pattern whatsoever, no rhyme or reason. And the resolution to the C value paradox was to say, well, if most of the genomes of organisms are junk that are just assembled through random biochemical processes, then we wouldn't expect any kind of relationship whatsoever between genome size and the complexity of the organism, because most of the genome is junk. And along those lines, people argued that one of the types of junk DNA classes in the human genome are something called pseudogenes. And pseudogenes are about 2% of the human genome, so they're a relatively small portion of the human genome. But for many biologists, the presence of pseudogenes, again, is this open and shut case for an evolutionary history to life. And I'll explain why they would say that in a minute. There's, again, roughly about 20,000 pseudogenes in the human genome, as well as about 20,000 intact genes. Now, to understand a pseudogene, we need to review very quickly the gene structure, and we went over this in detail in a previous episode. But in eukaryotic,
you're saying it's almost one-to-one, one pseudogene to one actual gene.
Yeah, yes, approximately. The numbers that people report are going to vary from paper to paper, and that a lot of times is due to the methodology that people are using to identify pseudogenes and things like that. So,
So when we say that 30,000 genes, these are 30,000 genes without the pseudogenes, or this includes the pseudogenes?
No, it's without the pseudogenes. And so then there's another somewhere between 10 to 20,000 pseudogenes in the human genome, again, depending upon how you would identify them and tabulate them and things like that. But it's roughly an equivalent composition, about 2%.
Amazing. Okay.
So with respect to the structure of a gene, remember we talked about the idea that in a eukaryotic cell, genes consist of two components, exons and introns, where the exons code for amino acids. For the amino acids, they code for the protein. Introns are these interrupting sequences, and that when that RNA is produced from that gene through a process called transcription, the primary transcript undergoes splicing so that the introns are removed and the exons are conjoined together, and then it's that processed message that winds up getting translated into proteins. So you have exons and introns, and you also have what's called the promoter, which is a region where the RNA polymerase binds, which it reads the DNA and produces the RNA transcript.
Yeah.
Okay, now when you look at the genome, there are a number of quote unquote genes that look like they have undergone some significant alteration through mutational events. Some of them are missing promoters, some of them are missing the start codon, others have undergone what are called frame shift mutations, where the reading frame is different. Some have been truncated where part of the genome or the gene is missing. Some have missing introns, and some have a premature stop codon, and the list goes on and on. And so the thought is that these were at one time functionally intact genes that experienced severe mutations that disabled them. And it turns out that there's a number of different types of pseudogenes that are found in genomes. And so one of them is referred to as unitary pseudogenes. These are actually fairly rare, where they involve a gene that's undergone mutation. And as a result, it's no longer capable, at least in principle, of producing a functional protein. And yet the organism is able to survive without the gene because it doesn't necessarily play a necessary role in the organism's survival. So the classic example would be the GULO pseudogene in the human genome. This codes for a protein that's involved in the metabolic pathway that produces vitamin C. And because we consume so much vitamin C in our diet, the fact that we can't produce vitamin C has no bearing on our survivability or reproductive fitness. Whereas, organisms that are not able to consume vitamin C would need to be able to produce vitamin C as part of their metabolic operations. And so because it's not necessary in that sense, it's you can survive with a disabled copy of that gene. Another process, another source of pseudogenes involves a duplication process where during replication, some kind of mistake happens and part of the genome gets duplicated. And these duplicated genes, again, represent an extra copy of the gene where you only need one copy of the gene. So the other copy undergoes mutations and it doesn't seem to have any kind of functional consequence other than producing a pseudogene that's a defective copy of the healthy intact gene. And then last but not least, there is a third category of pseudogenes called process pseudogenes, whereas in this case, once the RNA is spliced and processed, reverse transcriptase can convert that RNA back to DNA that then becomes reintegrated into the genome as, again, what's called a processed pseudogene. So these are the three categories. But what evolutionary biologists will point out is that we understand, or at least we think we understand, the mechanism for how these different types of pseudogenes form through random biochemical processes. And by the way, it turns out that many of these pseudogenes that we see in humans are shared with chimpanzees. And in fact, they are shared with other great apes as well. And by looking at the sequences, you can build these nested hierarchies that essentially reflect evolutionary trees or correspond to evolutionary trees that evolutionary biologists would build. And so the thought is that, look, this seems to be, again, clear evidence for human evolution. Why would a creator introduce these non-functional sequences into genomes at the same location and with a set of patterns that seem to reflect an evolutionary history? Clearly the creator, if he did it this way, must be deceptive, right? That's kind of the argument that you see. And so again, what I want to point out here is this, that as somebody who holds to a creation model / intelligent design perspective, I'm of the conviction that the genomes of organisms reflect the creator's handiwork. And so my orientation would be not to see things like pseudogenes as being useless junk, but really asking a different question. Why would a creator introduce these types of sequences into the genome, expecting that there's going to be answers, that there's going to be a rationale for why that's the case? But to legitimately interpret pseudogenes in that way, we not only need to explain why they're there, that is, what function do they serve, but we also need to explain the pattern of nested hierarchies, and also why would they be so similar to genes? Why would pseudogenes be so similar in structure. And it turns out that here we are 20 years after the human genome has been sequenced, and is now really, I think, a revolution in the way that we think about the human genome and different types of junk DNA, including pseudogenes, where we now have accumulated a lot of evidence that pseudogenes serve a wide range of functions. Now, I'll point out here at this point, and I think this is really a significant point, and I'd be interested in you, in your thoughts too, Ahmed, here.
Yeah.
And that is that, the discovery of function for pseudogenes was coincidental. It was accidental. People weren't out investigating pseudogenes. They just happened to stumble upon evidence for their functionality. And in fact, it looks as if this view of pseudogenes, shaped by the evolutionary paradigm, really got in the way of scientific progress. Whereas someone like you or I, who holds to a creation model perspective or an intelligent design perspective, would actually ask questions that would have accelerated the discovery of function for pseudogenes. And the reason I bring this up is that so often I hear skeptics say, if we just accept your ideas that there is bona fide design in biology, that it reflects the creator's handiwork, if you just say God did it, that just shuts down scientific investigation. Well, here's the opposite happening, where by holding to a materialistic view of origins, the scientific enterprise has been stultified. And in fact, when you read this review article written by a team of biochemists, published in Nature Reviews Genetics in the spring of 2020, they complain about that very point, that labeling pseudogenes as pseudogenes means that nobody's interested in studying them anymore. So I just think it's ironic that the complaint that many people have against kind of a creationist intelligent design approach actually is realized in an evolutionary paradigm.
Yeah. It is really. I totally agree with you. That once you label something as an error, there is no further need to look into it, right? So if from the beginning people are looking as this is designed and the designer is wise, so whatever is there must have a function, and now it is our role to find out about the function rather than to give it a false tag and then call it a day, then for sure this would have progressed these discoveries. But also I think that this is not only in the matter of pseudogenes, but in so many other things that are all over the place. The very fact that they call this the 2% that makes sense and the 98% that doesn't make sense, regardless what is the content of the 98%, means that science has stopped at the 2% just because of ignorance and having a paradigm where things have been built by a random process. And then you have a science that has only a 2% IPAN versus 100% IPAN. So we're looking at a 50-fold more discovery that would have been undertaken by somebody who is looking for wisdom in creation rather than by somebody who is looking for randomness and looking for the needle in the haystack that makes sense, that has come as the improbable probability that then caused things to happen since the beginning of life till the human being. So 100% agree with you. And something that is typically done in computer science and even in cryptography is that you try to outsmart your opponent by making part of the message look like rubbish, right? Yeah, so I want to send an encrypted message, and I give you the hard disk, and it has a program, and the rest is is the real purpose of the message, but I'm making it look like it is not. Okay, so what will I do? Number one, I will not put it, for example, in the directory structure. So you cannot find the file just by going to the directory, right? Or to the folder list, right? I will not put the typical markers that mark the beginning or the end of the file, etc., etc. But the receiver of the message will know where to find the information exactly. And like, for example, what you were talking about, I'm not sure if ERVs will be classified under pseudogenes or not, would they?
No, they're a separate category altogether.
Yeah, I will just use them as an example, for example. So for example, if I have an antivirus on my machine, okay, the antivirus needs to have the signature of the viruses that it's looking for, right? And it needs to have it in a way to the extent if you scan for viruses on your computer that has the antivirus, that has the signatures of the virus, then the antivirus will not mistake the contents of the antivirus itself as being a virus. So it needs to be stored in a way that does not lend itself. Number one, it doesn't want to activate the virus. And number two, it doesn't want to be scanned and identified as a virus. So sometimes you will need to code a certain message to the extent that it does a function, but you will need to distort the message in a way to prevent its side effects. Mm-hmm. So many of these techniques we actually use, whether in encrypting messages, whether in hiding messages. And sometimes we have to do that because otherwise the message might be harmful or disruptive or give false impression, but it still remains functional. Yeah. You just need to know what the programmer is doing, and sometimes it needs wits, and sometimes the programmer just intends it to be this way because right. It is part of the function of an encrypted message to be encrypted, let's say.
Right.
Yeah.
Right. Well, and I'm going to be very interested in light of your comments. I always learn so much from your insights, Ahmed. And again, it's mind-boggling to me how the principles of computer science and computer engineering seem to manifest over and over again in biological systems.
Yeah, I guess so.
But in this particular paper, the researchers identify at least 6 functions for pseudogenes, and there are probably many more that have yet to be uncovered. And part of the point of their paper is that we really need to investigate pseudogenes as very important features of genomes and set aside this bias against functionality. But for example, they point out that many times processed pseudogenes actually produce functional proteins. They actually are transcribed by the cell's machinery and produce functional proteins that are distinct from, quote unquote, the protein that they were, the protein encoded by the gene they were originally derived from.
Wow.
Also, they point out that sometimes.
So why, if it is actually transcribed, would they, in the first place, classify it as pseudogene? And how does the cell manage to transcribe it if it has those missing elements or frame shifts, etc.?
Right, and well, for example, it very well could be that there are promoters that are still associated with it, or that sequences upstream can function to some degree as promoters, and we're not recognizing them as such. But part of it was initially looking at the human genome sequences and just saying, this looks like a processed pseudogene, it must not be functional. So, and then when techniques were developed in support of the ENCODE project, where people were surveying the genome for functionality, they were discovering a lot of transcripts. A lot of regions of the genome were actually being transcribed that initially they didn't realize were being transcribed, including processed pseudogenes. And so there's a growing number that are recognized as being transcribed and translated into useful protein products. Part of it too is that when people are measuring transcription, because of the similarity between pseudogenes and the quote unquote intact genes, many times when they detect pseudogene transcripts, they assume that those transcripts are actually for the intact gene. And so some of it is a methodological thing. But, more and more, I think people have an eye towards the fact that these processed pseudogenes very well may be transcribed and translated, and are finding a large number of examples where that's the case.
So why can't we actually reframe the problem as like pseudogene is a misnomer, actually? And maybe we should have, it is just because we expect genes to look in a specific way, then whatever does not look according to our initial understanding of a gene, we call it something else. But really it is gene type 1 and gene type 2, or category 1 and category 2. It is just that we call category 1 gene and category 2 like junk. And then we classify junk into different kinds of junks, ERVs, pseudogenes, this or that. Yeah.
Yeah. And again, part of this review article, and the scientists writing this are not necessarily friends to our perspective, right?
Yeah.
But they're basically saying that this is really hampering, that this misclassification, they're making the very point that you're making. So,
oh, really?
Yes, exactly. So
wow.
Now, what's also interesting is even genes that have been quote unquote truncated pseudogenes can still code for useful proteins. And the reason why is because, remember, we talked about the fact that in proteins there are domains, where in the three-dimensional structure there are these discrete independent units that are connected together, where the units perform distinct functions. Well, many times with truncation, you still have functional domains that are present that are now going to serve a different function as a truncated protein versus the original protein. And so again, you could say, well, maybe the creator intended it this way. And maybe the creator has a category of genes in our genome that don't have introns because that impacts something about the gene's biochemistry that we have yet to fully appreciate. That maybe having genes that have no introns and genes that have introns are critical to accomplish very different functions, but nobody is even asking that question because of this evolutionary bias that people have. But the fact that we're seeing functionality here should suggest that maybe there's an advantage to not have introns, or there's an advantage to have introns, and so we aren't even asking those questions.
Yeah, I think even the matter of introns that we discussed in the splicing episode, that I would really suggest that audience will get back to it if they didn't see it. Even the matter of introns was thought of in the beginning like as junk and having no value, and then all those great benefits of having introns unfolded, and as it is like a series of ignorance, or a circle of ignorance, where number one, you assume that even the gene, it has parts that have no value. And then when you even don't find that part, then you consider it to be a pseudogene. The whole paradigm is built on a specific mindset where you are expecting only a little piece of a little piece of a little piece to have meaning, and the rest is just junk, because you are building your perspective that everything is just randomness, where only the improbable slight probability has happened in this universe or this planet or this strike of luck or something.
Yes. Now let's move to the middle panel. This function involves gene regulation. So when it comes to the DNA double helix, right? Remember, we talk about the complementary base pairing rules, so that if we know the sequence of one strand, we know the complementary sequence of the other strand, because adenine always pairs with thymine, guanine always pairs with cytosine. So when a DNA double helix unwinds locally so that the RNA polymerase can read that region of the DNA as a gene to produce the RNA transcript, there are, in a sense, two strands that are exposed, but only one of them is read by RNA polymerase. That's called the sense strand. And the other strand is called the antisense strand. Well, it turns out that if you actually transcribe the antisense strand of pseudogenes, you now have an RNA transcript that's complementary to the primary RNA transcript that's being produced by the quote unquote intact gene. And that causes them to actually form an RNA double helix that then essentially silences the translation of that transcript. And so one of the functions of pseudogenes is to provide that antisense RNA to cause RNA interference, preventing the translation of RNA after it's being transcribed. So in other words, there are two levels of gene regulation that are being exposed. One is by turning on or turning off the gene or regulating how much of that RNA is produced. The other is by breaking down the RNA before it gets translated. And so there are these competing processes that are going on, which is called a biochemical feudal cycle. And there are good reasons why you would do it that way, but that is beyond the scope of our conversation today. But nevertheless, the pseudogenes are serving a role of providing RNA interference, preventing the translation of RNA transcripts. This now explains why pseudogenes are going to look similar to the intact gene sequence to allow for that base pairing to take place. It also means that if there are mutations, or maybe we should say changes in the nucleotide sequences or differences in the nucleotide sequence of the pseudogene compared to the intact gene, the assumption is that those differences are due to mutations. But what if they are deliberately introduced to either increase or decrease the affinity of that RNA transcript for the primary transcript? So you can begin to see that maybe some of the differences aren't due to mutations, but are intentionally designed to accomplish a type of fine-tuning to the RNA interference phenomena.
Wow.
Now,
that's amazing.
The next function has to do with the pseudogene transcript functioning as a molecular decoy. And I'm going to spend a little bit of time unpacking that. This is called the competitive endogenous RNA hypothesis. But suffice it to say is that what happens is that the pseudogene actually functions as a molecular decoy, and so that the cell's machinery will break down the pseudogene transcript, allowing the intact gene transcript to be translated. So it's a decoy mechanism.
Can you go again? Can you explain that again?
Yes, and I'll unpack it in detail. But the idea is that if both the pseudogene and the intact gene's RNA is being transcribed, and part of the process of gene regulation is breaking down RNA transcripts once they form. If you want that intact gene to be translated at a high level by expressing the pseudogene and getting the high levels of pseudogene transcript, those pseudogenes are going to be consumed by the degradation mechanism, not the intact gene, and it allows the intact gene to make it to the ribosome to be translated. If you shut down the transcription of the pseudogene, it means that the intact RNA is going to be destroyed before it can be translated. So it becomes a decoy mechanism, and by regulating the level of pseudogene transcription, you can regulate the level of protein transcripts. And I'll give you a concrete example of that in a minute.
Yeah.
But this is another example of a gene regulation mechanism that is utterly dependent upon the similarity of the pseudogene sequence to the gene sequence.
Okay.
And then last but not least, there's another function. People have observed that if you splice out the pseudogene, it actually causes the chromatin structure to become reorganized in such a way that shuts down gene expression. So there are some pseudogenes that are never transcribed, but they seem to provide some kind of structural stability that nobody quite understands, structural stability to the chromosome. There's other studies that have shown if you remove the pseudogene sequence, it increases the rate of what's called non-allelic recombination, and this creates all kinds of havoc for the genome. It destabilizes the genome. And so the point here is that some of these pseudogene sequences seem to be serving some kind of role of imparting stability to chromosome structure that's not fully understood. So even if it's not being transcribed, that pseudogene very well may be performing a function. So the point here is that we are now seeing a wide range of different types of functions that pseudogenes are performing, many of which are actually dependent upon the identity, the similarity in the sequence of the pseudogene with the intact gene. That's kind of the point. So now you were asking about the decoy mechanism. And so that decoy mechanism has actually been generalized into something called the competitive endogenous RNA hypothesis. Now, I'll explain what this is in a minute, but this is really significant because we're going from actually saying, oh, there's function to pseudogenes that we didn't previously realize, to actually now developing a theoretical framework that provides a fundamental explanation for the presence of pseudogenes in the genome. So it's one thing to say there's function, but if I can actually now develop a model or a hypothesis that gives a deep-seated rationale to why pseudogenes are present, that suggests something far more than just simply, the haphazard evolution of function from something that may have been non-functional at one point. It actually suggests if there's a rationale, if there's an organizing principle, that's what you'd expect the mind to be responsible for. And so that's the point that I want to make there. And so the idea behind the competitive endogenous RNA hypothesis and this idea of a decoy mechanism is kind of illustrated, I hope, in this slide. And what's being shown here is that RNA is being transcribed by RNA polymerase. And there are two fates for that RNA. It can either be translated at ribosomes, or it can be broken down before it gets translated. And what happens is that there is a protein complex called RISC, R-I-S-C, that will bind to RNA transcripts. And if, in addition to that, a microRNA, a small RNA, binds to the RNA transcript at what are called microRNA recognition sites, it'll cause another protein called AGO2 to bind to the RISC complex and break down the RNA transcript. So it's all dependent upon these microRNA recognition sites in the 3' untranslated region of the RNA. And so this is showing for 3 different RNA transcripts the untranslated region, and there are different colors here that correspond to different microRNAs, that these are microRNA binding sites. And so the point here is that, depending upon what, MRE is present in the untranslated region of the messenger RNA, that's going to determine which particular class of microRNA is going to bind, right? But there's
what's MRE again?
MicroRNA recognition elements. And so in other words, in the RNA transcript, there are places where very small RNA molecules called microRNAs can bind. And when they form that local duplex, that then becomes the recognition for the RISC and AGO2 complexes to basically break down the RNA.
Okay.
But here's the point. Because there are different microRNA binding sites, it means that if RNA molecules have the same MRE, microRNA recognition elements, then they're in the same. They're considered to be in the same transcript family.
Yeah.
Right? So there are families of RNA transcripts where the membership in that family depends upon again, what kind of MREs you have in the untranslated 3' region. Does that make sense?
Yeah.
Okay.
So if I'm a risk and I'm looking for an RNA strand to decompose, and there is another one that looks like it, I might as well go to the other one that looks like it. If
Right.
there are more RNA strands to be decomposed than risks, so I'm
Right.
expecting that those pseudogenes would double up to keep the risks busy for
Right.
some time or something.
Right. And
Yeah.
so here's a concrete example of what we're talking about. This is one of the very first studies to recognize pseudogenes as being decoys, molecular decoys. So on the left-hand side, this is a gene that codes for a protein called P10, which is a tumor suppressor protein. And the researchers discovered that and there's a P10 pseudogene. They discovered that if you eliminated the pseudogene from the genome, this was in a mouse model, the mouse developed tumors very quickly. They were prone to tumor formation. As soon as they added the pseudogene back, it shut down tumor formation. It led to tumor suppression. And so they realized that the pseudogene transcript and the intact gene transcript both had the same microRNA recognition sites. And so what that meant is that when the pseudogene was transcribed, it became a decoy where it was destroyed, allowing the P10 transcript to go to the ribosome and produce the P10 protein, which led to tumor suppression. And so the competitive endogenous RNA hypothesis is essentially generalizing this discovery by basically saying that this could be actually a unifying principle for why pseudogenes are present in the genomes of organisms. And again,
that's amazing. I just have a little question here, because some of the audience will think, so why are there risk elements and ego proteins anyway that are just going around and destroying RNA transcripts that need to be translated? So
this would be an example of what biochemists call a feudal cycle. And so the classic example of a feudal cycle is seen in the glycolytic pathway, where you have an enzyme that converts a fructose 6-phosphate into fructose 1,6 diphosphate or bisphosphate. That step is an energy consuming step in that metabolic pathway. There's an enzyme that basically removes the phosphate from fructose 1,6 bisphosphate. It removes it going back to the starting point. And so biochemists have wondered, well, why would the biochemical pathway produce an intermediate that's very energy intensive to produce, only to break it back down and revert it back to the original metabolite, right? The original metabolic intermediate. That's called a feudal cycle, where the two enzymes are offsetting the activity of one another.
Yeah.
Well, it turns out that when you have a feudal cycle, by slightly adjusting the activity of one or the other enzymes, you can actually dramatically increase the flux through the metabolic pathway, compared to if you didn't have the feudal cycle, where you only had one enzyme that you could manipulate its activity. And so it's counterintuitive, but when you work out the math for the enzyme kinetics, you actually, through that feudal cycle, have a very sensitive lever, so that if you just tweak it slightly, and boom, the flux increases dramatically through the pathway. And I suspect that that's what's going on with transcription, is that you're producing transcripts, which is energy intensive to do, only to have them destroyed. Well, that doesn't make any sense. Why not just prevent them from being produced to begin with? Well, I suspect what's happening is that through that feudal cycle, you're actually creating scenarios where by tweaking the production and the destruction of the RNA transcript, you can dramatically increase the flux through that pathway, allowing you to either shut down dramatically or increase dramatically protein production when it's needed. So I
Yeah,
think that's the
actually,
explanation.
yeah,
Go ahead.
that's very insightful. Actually, in electronics, you have these things called negative feedback loops,
Right.
and stable systems need negative feedback loops. And what negative feedback loops allow you is to have this control logic where you will take the output, put it through an amplifier or a gain function, and feed it back to the input. So you have like a dial, or you have a toggle where you can, in the input, there is always a plus and a minus, where you're taking the output, and like you have the cruise control in your car.
Yeah.
What the cruise control is doing all the time is that it's measuring the speed of your car, and it's feeding back to the steering to increase or decrease. And what that enables is that if you want a set speed, then the cruise control, if it finds you, for example, going down a ramp, it will find that you're accelerating, and then it will add a deacceleration. And if you find that you are deaccelerating because you're going, for example, up a ramp, it will add back acceleration to go back to the set point. But what it also causes is that if you click the dial of the cruise control to increase the target speed, then this very mechanism can now increase the flux. It will then see that the target is higher and cause acceleration, and vice versa. So the very existence of the negative feedback loop causes two things. Number one, you have stability because you can have a set point and the system will automatically converge to the set point. And the second thing is that it gives you control because you can actually set the set point. So, and that's actually why on cruise control, you always have like this, like set and increase and decrease and coast and this kind of controls. And this is only possible if you have a negative feedback loop. While on the other side, if you have a positive feedback loop where more output means even more output, the system is typically unstable and will crash. And you can notice that when you are talking in a microphone and there is a speaker. And then what happens is that the output of the speaker goes into the microphone and then back to the speaker and then into the microphone. And then what you will find is that and then it will cut off. Because at a point, there will be a circuit breaker that causes the system to reset. So a positive feedback loop will cause a significant increase, a total runaway output, and then a crash. So, it's just so amazing how these things are intrinsically built in biochemical systems to the extent that then researchers should look for who is setting the preset point and what will cause something to cause, for example, more tumor suppression or less tumor suppression, maybe according to the status of runaway mutations or being exposed to an environmental cause that is destroying the animal or something, and then more tumor suppression is needed or less tumor suppression is needed, for example, in your example. And I'm sure that there must be some research that is looking for what is actually bringing this system to certain specific set points.
Yeah, and you know, I thought, yeah, your discussion is fascinating, and you know, I just think that there's what impresses me is just again how elegant the design of these systems are. That there's a rationale for everything. That when you ask the why question, there's an answer, right?
And there is typically an answer that you can find in the smartest systems that we build.
Yeah.
Typically, that's amazing.
Yep. Okay, so now remember I said that that again RNAs that have the same MRE sites become part of the same family, and so what you now have are these transcripts, these RNA transcripts from different genes and pseudogenes that are forming this network of transcripts where the transcripts are all, you know, part of the same family. And because some transcripts have more than one microRNA recognition site, they actually are involved in multiple networks, right? And so it's a very complex relationship now among these RNA transcripts. But what that means is that when you increase the microRNA level for one particular MRE, binding site, you're going to, through that activity, influence the level of all the transcripts that are in the family.
Wow.
Likewise, if you upregulate the expression of one RNA molecule that is in that network, it's going to cause all the RNA molecules to be translated at a higher level. If you reduce the amount of a single RNA molecule in that network, it's going to cause all the translation of all those protein products to be downregulated as well. And so now you've got, and so it's kind of like this very complex set of decoy mechanisms that are at play, where, it's kind of like boats on the ocean, whereas the sea level rises and falls, all the boats rise and fall together. And what that means is that it explains why you would have unitary pseudogenes in genomes that could actually be transcribed, even though they're never going to produce a functional protein. They still are playing a role in the network in this RNA
competitiveness network.
In this network, and they're still functioning as part of this decoy mechanism. So this idea of this competitive, endogenous RNA hypothesis essentially means that you've got, again, this incredibly elegant mechanism for regulating gene expression that has gone really unrecognized until recently. And as you're saying, it adds a level of sophistication to gene regulation that produces stability and also allows for a very precise control and a very rapid response time to changing environmental conditions, allowing proteins to be produced or their production to be shut down very quickly when those proteins are no longer needed. Wow. And so, to me, when we start thinking about what we've discovered about pseudogenes, instead of this being a liability for a creationist or an intelligent design proponent, or instead of trying to simply stave off the challenge from the evolutionary paradigm, we should go on the offensive and highlight the fact that this is an incredibly ingenious, elegantly designed regulatory system that a creator has put in place that that we should marvel at as opposed to anything else.
Yeah, absolutely. It's amazing the way you're explaining that there is not only just one feedback loop, but there are actually so many feedback loop interlaced together. And in our engineering, we don't do that. It's too complex for anyone to design, actually. So this is where now there is great gap. There is a leap between what the most wise creator can design and what we can design. And I think then it will go to biomimicry now. If maybe we need to learn something about feedback loops and control systems from this amazing design that we did not even touch the surface. Compared to it. Yeah.
And so what we've done up to this point is we've basically said, well, there's a rationale for pseudogenes. They're functional. There's a rationale for why their sequences are similar to that of quote unquote intact genes.
Yeah.
And we could just simply say that the fact that these sequences are shared makes perfect sense if they're functional, because then we would see this as common design features, not an indication of common descent. And so we've satisfied the three criteria that we need to satisfy in terms of explaining pseudogenes in a coherent manner, from a creation model perspective. And the fact that you can form these nested hierarchies, to me, just simply again suggests common design. Things that are designed you can organize them
them.
into, in like, a very simple, maybe silly example would be silverware, where you have, butter knives and steak knives and, soup spoons and teaspoons and tablespoons and dinner forks and salad forks, dessert forks, and they all have certain features that are in common, but also certain features that make them distinct. But you can, all the silverware that belongs to the same pattern would form the largest circle, and then the knives would form circles, and the forks and the spoons, and then within there could be even further subdivisions. And so there's a nested hierarchy where the similarities and the differences reflect some aesthetics, but they also reflect the functionality and the purpose of the different pieces of silverware, and the differences aren't due to random events, but are intentional differences. So why not think about genomes in the same way, where the sequences that are even if they are in the form of nested hierarchies, still actually may reflect, intentionality, that that the shared features reflect commonality in their function, and the differences reflect, again, uniqueness in their function with respect to the role they're playing in different organisms. So the point is that there's a lot to marvel at when we think about pseudogenes from a design framework.
Absolutely. And is there an answer yet to the matter of similarity in position?
That is a question I don't know. The sequence of genes along a chromosome is referred to as its synteny, and there very well could be a reason why, certain genes are adjacent to other genes, and why, and there could be an importance to their precise location. I don't know what that would be, but this again is where I think the framework really makes a big difference. From our perspective, we would say we don't know the answer, but we are going to suspect that there's a reason for this, and we're going to be asking, certain questions that'll actually drive scientific advance. From an evolutionary perspective, you would just simply say, this is just what evolution produced without any rhyme and reason, and just stop there, right?
Yeah. From what you said, I thought that the matter of structural integrity or helping chromatin to maintain certain shape or make sure that things do not get suppressed or muted in a way where there is interaction with histones, whatever. Might that be actually a clue to where the pseudogenes should actually be? Because we've tried excising them out, but what if we have tried actually moving them around? Maybe other problems would have happened, because in this specific place, it also has a structural role. Maybe this structural role is as important in humans like in chimps, and hence their phenotypic similarity would necessitate that these things are in the same places, etc. What do you think about that?
Yeah. I if I was pressed to give an answer to your question, my answer would probably be very similar to yours. I think that's the first reasonable place to go in an scientific inquiry into that question. And there may be other reasons that we're completely, oblivious to, that if we found the answer, we would even, marvel further at that insight. But this is what makes things exciting to me as a biochemist who sees God's handiwork in biology, is that, there's just a lot of excitement as to think about, why would a creator do it this way, with the expectation that I'm going to be surprised by the answer, right? In a way that causes me to marvel, and likely in a way that might even inspire a new technology. And so there's a lot of reason, from our worldview perspective to engage in these kind of scientific investigations with excitement and joy and an intensity of looking for the answer. I've always felt that evolutionary biologists have such a myopic view of biological systems. It's such a depleted, impoverished view of biology, and that to me is just so unfortunate that someone would devote their lives to studying biological systems, but have such an impoverished view of their elegance and their beauty. What a sad way to look at biology, I think.
Absolutely. And I think even there is a very important clue there, or actually the starting point is we'd better understand synteny first before we complain about pseudogenes being in the same place, because we don't know what the place means anyway. So yeah, so also a brilliant point that you have actually pointed out too. Yeah. All right. So where do we go from here? We still have some presentation to run, right?
No, well, I was just going to go ahead and stop here. I think I made the point that I was going to make. So I think we're good.
All right. So I'm going to cut this part off, but if you think there is more to it, we can actually continue recording this next time.
No, I think we're good. I think we've made our point. So
yeah. Okay. So let's have our conclusion discussion here. So I think this is one more example where, as you said, and we discussed a few times before, systems that are designed with a great intelligence behind them will not be shy from answering the why question. Why is this way? Yeah. And systems that are random would be, well, there's no answer. They're random. You can expect anything. But systems that are actually designed and magnificently designed with proper effort and proper marveling into them will actually at the end answer the why question and open new horizons and new areas for medicine and new areas for advancement and human wellness, etc. So I think this has been a great example to that. Do you agree to that?
Yes. Yeah. And in fact, that the review article that I pointed out earlier, part of the point that the researchers make is that pseudogenes now become therapeutic targets.
Oh.
And think about that. I mean, apart from recognizing their functional importance, you have shut down an entire area of biomedicine that could potentially lead to treatments and therapies for diseases in that currently don't have those treatments available. So again, it's just another example of how a materialistic approach to biology really can potentially get in the way of actually making very important progress for humanity's sake.
Yeah, I guess this is a great closing line here, especially that this episode might be, let's say, the end of our part one of this series, or the second part of the first season of this series. Let's say that adopting a model where you are looking at life as being created by a wonderful and magnificent and wise creator is to our benefit, is to the benefit of humanity. It makes, number one, it makes science fun, because if you're looking for something, you're going to find it. And number two, whenever you ask the question and you find the answer, you are better health, better wellness, better understanding, and definitely stronger faith. So why would somebody choose the alternative path if it is, number one, boring, number two, futile, number three, over and over again, proves that the system is behaving as a created system, not as a random system. And it is at the end to our benefit in this life and hereafter. And what are you going to lose if it's a winning streak all over the place? So I guess, yeah. So yeah.
Yeah, I couldn't say it better myself. And so
well, Fuz, you've already said it in a better way. So I guess myself and all the audience are so grateful to your explanation today. I think this topic of pseudogenes and the other topics that we have gone through, for those who have watched this series until this episode and who are definitely excited to find more to come, I think this has been mind-boggling, intriguing, satisfying for the faithful, satisfying for those who are looking for science, and satisfying for those who are looking for unmistakable evidence that this is a world that has been created by God, the necessary being who has created it and is maintaining it, let's say. So wonderful work. Thank you very much, Fuz, for those wonderful presentations. And how about your comment for this, I would hope, an intermittent pause for this series that needs actually to go on? What would be your comment for what we've gone through?
There's so many topics that are left to be explored that I think we're going to have to do a second season. So
I would definitely agree. So I'll take this as a promise. But what would be your comment at this point? What's a message that you would like to briefly give to the audience?
Again, I think it would be, what we like to say at Reasons to Believe, which is the organization that I work for, is that the more we discover about science, the more that we have reasons to believe. And so this idea that science is somehow antithetical to faith is just so far off base that if we really are looking at the world around us, including, of course, living systems, and we just, if our eyes are attuned to it, we just see so much evidence for the Creator's handiwork. And there's a real beauty to the creation, not just simply the beauty of a sunset or the beauty of an ocean expanse, but when we roll up our sleeves and begin to understand the inner workings of things at a scientific level, we don't make the beauty of the world around us clinical, right, through science. We actually amplify the beauty in the marvel of creation. And so, to me, I think for people of faith, people that are, that view God as a personal, transcendent creator, that science should be something that we pursue with great enthusiasm, never ever losing faith that as we learn about systems around us, if we think about them properly, they're going to point us to the Creator.
Absolutely.
And to me, the study of biochemistry is an act of worship.
Yeah. So I'll say the one word that comes to my mind just going through what we've gone through is Subhanallah in Arabic, which is God is so exalted, or exalted is God. Yeah. And it reminds me of also one verse in the Quran that says, the translation would be, we will show them our signs in the horizons and within themselves until it is evident to them that it is the truth. Yeah. And I think this is exactly what we've been looking at. Definitely within ourselves, but also within every organism, and definitely in the horizons of science we're standing to find more marvels over the time. So, Faz, God bless you.
God bless you too.
30 trillion thanks, and each one of them has 6-point-something billion letters or something for this great presentation and this great series thus far. And I'm sure all of the audience are looking for a season 2. So until then, God bless you, and God bless you all who are watching, and peace be upon you, Faz, and everybody. So for everybody, see you again soon, and don't hesitate to put some questions, and we will be looking into making maybe an after show once we collect enough questions to give you a collection of answers to this one and the previous ones too. So peace be upon you all, and see you again soon.